Hello everyone, I'm Mark Tugay, and our lesson for today is about signal transduction, specifically on cell cycle regulation in the cell. So in this lesson, we'll learn and understand the cell cycle regulation in the cell, specifically illustrating the cell cycle, explaining how the cell cycle is regulated in the cell, and describing the importance of the cell cycle regulation in the cell. So first, you will be assessed, okay, on your prior knowledge regarding the topic through administering the pretest using Kahoot. So let's take the pretest. Right? So this is a student-based test. So there are 10 items. Oh, ooh, still. Ooh. Oh no. Ooh. Two more. All right. Got it. Okay, so it seems like I need to read more, okay, to understand, to learn about the cell cycle regulation in the cell. So, but no worry if ever you will be getting the same score as I had. So, what is important is at least you have checked how much you know about the topic and what is important later on. And I do hope that after the teaching learning process that we'll be having, okay, will make a difference, okay, completing the assignments and other activities that will be given will make a remarkable difference when you will be tested again for the post test. So to start, I would like you to log in on your Quizlet account for you to explore and unlock unfamiliar words and try to other assessment tools or activities to enrich your vocabulary regarding our lesson. So, which definitely helps you in understanding the different processes involved in the cell cycle regulation in the cell. Okay, so let's see what's in the Quizlet. So I have prepared, okay, so using this online tool will help you practice and master what you are learning specifically regarding our topic. So Quizlet has seven standard study modes so that help you review in the way that works best for you. So we have here the flashcards, a learn, write, spell, test, and we have also here games or that they can play with. So match, mix and match, then gravity, and live like the Kahoot. So the flashcard is basically a quick way to review terms and definitions, just like paper-based flashcards. So you can see here the vocabulary words and the definition if you tap it. Okay, then the next. So again, word and the definition. And learn. So it's basically like a multiple choice test. So we have the definition, we have here different choices. Then for the write, so you're basically writing the word that is being described or the other way around. Given the word, you have to write the definition. Okay. Then next we have the spell. So basically it is a way to study mode that it dictates a term or definition, then you must correctly type. So spelling at some point will be considered. So test, so different types of tests, multiple choice, okay, short answers, okay, matching, okay, then the different activities. So again, you are given 10 to 15 minutes to familiarize yourselves with the different words presented. And again, you can have it as well later on at your own pace to enrich your vocabulary words and to understand, okay, and it will help you for our lesson. So to know more about the cell cycle regulation in the cell, so let's study this first figure. So the cell cycle shows a series of events, okay, that takes place in a cell as it goes and as it grows and divides. So this involves two phases. We have interphase and M phase. So interphase is a preparatory phase that allows the cell to grow, carry out cellular functions, replicate DNA, and make sure it is ready for the next stage of the cycle. These activities are completed in the different sub-phases, such as G1 or GAP1 phase. Okay, it has the longest duration. It takes 6 to 12 hours. Cell grows and performs normal functions. In this phase, the cellular contents, excluding the chromosomes, are duplicated. The RNA and protein synthesis will also occur in this phase. So for the S phase, so it has a duration of 6 to 8 hours. Then DNA replication occurs where the cell copies its DNA in preparation for cell division. Then the third, we have the G2 or GAP2 phase where it prepares the cell for mitosis, and it lasts for 3 to 4 hours. So mRNA, RNA, and protein synthesis continue and takes inventory, making sure it is ready to continue to the next phases. So when these activities are completed, the cell proceeds to mitosis and cytokinesis, which lasts only for an hour. Mitosis is divided into four phases. We have the prophase, metaphase, anaphase, and telophase. And in mitosis, the cell undergoes karyokinesis, or the nuclear division. And its nuclear material separates into opposite ends of the cells, which is completely divided during cytokinesis, or the cytoplasmic division, where identical daughter cells are produced. So some cells, like the cells of the kidney, heart, and liver, to name a few, come out of the cell cycle after remaining in the G1 phase and enter the G0 phase, also known as the quiescent phase or the non-dividing stage. The cell goes here, it receives, if it receives signal to differentiate, or when resources are insufficient to grow and divide. If the cell satisfies these deficiencies, then it will return back to the cell cycle. So in order to maintain the integrity and fidelity of the cell cycle, there are checkpoints that play a vital role in coordination between different phases of the cell cycle. So they prevent the entry into the next phase of the cell cycle until events of the preceding phase have been completed. The checkpoints are stages in the cell cycle at which the cell examines internal and external signals and decides whether or not to proceed with the cell division. So figure 2 shows the cell cycle checkpoints, whereby there are two categories. First is the DNA damage checkpoints, um, and the second is the spindle assembly checkpoint. So the first DNA damage checkpoints, they function for G1, okay, S, and G2 phases of the cell cycle, named as G1 checkpoint and S phase checkpoint, and the next one is the G2 checkpoint. So these checkpoints ensure that damaged DNA is not replicated and passed on to daughter cells. So in G1 checkpoint, the cell checks internal and external condition are right for division, specifically assessing the cell size, if it is already large enough to divide, the nutrients, if cell have enough energy reserves or available to divide. Another is molecular signal. If the cell receiving positive cues such as growth factors from neighboring cells, and the last is the DNA integrity. If any of the DNA damage. So for S-phase checkpoint provides continual monitoring of the integrity of DNA to ensure that damaged DNA such as incorporation of incorrect bases or incomplete DNA segments is repaired before replication. Then for G2 checkpoint, it also continually check the DNA integrity. It will for proper DNA replication and prevents the initiation of mitosis if the cell contains DNA damage. Because such damaged DNA activates a signaling pathway that leads to cell cycle arrest in order not to create abnormality by the end of the cycle. So second, we have the spindle assembly checkpoint, which maintains the integrity of the genome occurring toward the end of mitosis. So this checkpoint monitors the chromosomes alignment on the mitotic spindle, ensuring accurate distribution of complete set of chromosomes to daughter cells. The failure of a spindle assembly checkpoint arrests metaphase, where the chromosomes do not separate until complete complement of chromosome has been organized for distribution to each daughter cell. So these are the checkpoints and what they do for the cell. So another important facet in the cell cycle regulation are the regulatory molecules that turn on or off the cycle. So these regulatory molecules, cyclins in particular, control the activities of cyclin-dependent kinesis or CDKs, affecting different events of the cell cycle, which include DNA synthesis, mitosis, and cytokinesis. So table 1 shows the cyclin CDK complexes formed during cell cycle regulation and their functions. So in the G1 phase, so it is regulated by cyclin D combined with CDK4 and CDK6, forming a cyclin G1 CDK complex that signals the cell chromosome replication and inhibits RB protein. So RB protein is an essential CDK substrate that participates in a mechanism that arrests cell division in G1 when DNA damage is detected. So in S phase, it is regulated by cyclins A and E, okay, each combine with CDK2, forming cyclin S-phase CDK that activates DNA replication. And last, we have the in M phase, it is regulated by cyclins A and B combined with CDK1, forming cyclin M phase CDK complex that activates mitosis. So all the checkpoints require the services of protein complexes, whereby their levels are increased when damaged cells are detected. This will cause the blocking of the cell cycle in order to have time repairing damaged DNA. One specific protein that senses the damaged DNA is the P53, which has the capacity to stop progression of the cell cycle, particularly in G1 phase, by blocking the activity of CDK2 until damage can be repaired. If the damage is so severe that it cannot be repaired anymore, then the cell undergoes program cell death or apoptosis. Okay, so figure 3 shows the cell cycle regulation and overall mechanism. So cell cycle progression is highly regulated process orchestrated by numerous checks and balances to ensure the division and proliferation is a favored outcome. Progression is mediated by cyclin-dependent kinases and their partner cyclins. Each pair is associated with certain phase of cell cycle. Cell cycle transition are regulated by cyclin-dependent protein kinases, protein phosphatases, and ubiquitin protein ligases. So in G1, CDKs and G1S phase CDKs activates transcription of gene required for DNA replication that prepare cells for S phase. And in S phase, it is initiated by SCF or the SKP colon F box containing complex ubiquitin protein ligase attaches S inhibitors of S phase CDKs, marking them for degradation of proteosomes. The S phase CDKs are active. CDKs activate DNA replication. So during in G2, okay, so mitotic CDKs induce mitosis, whereby during prophase, the nuclear envelope breaks down and chromosomes align the mitotic spindle, but they cannot separate until the anaphase promoting complex or APC-C, a ubiquitin protein ligase, attaches the anaphase inhibitor protein securin, marking it for degradation of proteosomes. After the chromosome movement to the spindle holes, the APCC combines with mitotic cyclins, causing the degradation of proteosomes. So the resulting drop in mitotic CDK activity, along with action of protein of phosphatases, results in chromosome uncoiling assembly of nuclear membranes around the daughter cell nuclei and cytokinesis. So this is the cell cycle regulation. So to have a quick check on your understanding about the lesson presented, I would like you to read the information in this image and give your insights. Then after that, I would like you to answer the question posted: why is cell cycle regulation in the cell important? So you can have take this for 10 minutes. Okay, so in this lesson, it is presented that the cell cycle regulation is crucial for proper cellular homeostasis. Communication between or within a cell is done through cell signaling, and change in the activity of the cell is sent as a signal that may trigger a cascade of reaction for the body to respond accordingly. And the physiological characteristics of a cell are determined by networks of interacting proteins that process energy, material, and information. Progress through the cell cycle is a sequence of divisions between the stable recurrent states of the three subsystems of the regulatory network. So the G1, S, GS, and the mitotic components. So passing these division points is driven by cell growth and progress from one stage of the cycle to the next can be restrained by checkpoint mechanisms that monitor the state of the cell's DNA and mitotic apparatus. If there are problems, so these checkpoint mechanisms push the bifurcation points to larger size and thereby halt or delay progression through the cell cycle. Okay, to end, I would like you also to write your summary of what you have learned about the lesson presented. So maybe you can have it 2 to 3 sentences at least for your summary. Okay, so and again, after the lesson, you will be tested. Okay, so post-test will be given. And like what I have mentioned earlier, I hope that at least there will be a remarkable difference when in taking the post-test after giving some of the activities. Okay, so I do hope that you will have an improvement of what you have learned in this lesson. So this ends the presentation. Ends our lesson. Mahalo everyone for listening.